everybody is familiar with biological weapons of mass destruction, WMDs. There was a very large infrastructure decades ago for weaponizing these pathogens, some of which you're familiar with, Yersinia pestis, which causes the plague, smallpox, okay. Let's see, what else? Bacillus anthracis, which causes respiratory anthrax. Francischella Tularensis, all right, that's one people may not be familiar with. Hunters are sometimes familiar with it because it's commonly known as rabbit fever. It's a bacterial pathogen. It is highly infectious. It takes very, very few bacteria to infect an individual, and it was weaponized by both the former Soviet Union and the US military decades ago. There is currently no vaccine available to protect individuals against it, military personnel or first responders or the general public. And this is a pathogen that is highly virulent. It's very easy to cause disease. There's very high mortality associated with it. If 10 individuals were infected with Francischella Tularensis, which causes a disease known as Tularemia, anywhere from 3 to 6 individuals would die from that infection if they weren't treated within the first 24 to 48 hours of diagnosis. So it's a very insidious pathogen. It's very easy to aerosolize. So you could envision someone in a crop duster contaminating a large area by aerosol distribution of a pathogen like this. And so this pathogen, not surprisingly, is considered a Category A select agent. So the CDC has designated this a select agent that has to be used under biosafety level 3 facilities. And all of the individuals working in such projects have to undergo very extensive FBI clearance to work with these select agents. And the Trudeau Institute is one of the few, if any, facilities here in the North Country that has the research infrastructure to carry out CDC-approved select agent work with pathogens like Francisella, like Yersinia pestis. And we are actively trying to develop the next generation of vaccines against these Category A select agents, these biological WMDs. Some of the most significant progress we've made just recently is through collaboration with colleagues at Clarkson University. And this is in the area of trying to develop a next generation of antibacterial agents to kill Francischella tularensis and other bacteria that have developed antibiotic resistance. So right now, the most pressing concern here in the United States for public health in general, particularly in civilian populations, is the spread of antibiotic resistant bacteria. And Francischella tularensis, particularly weaponized forms of it, are very resistant to antibiotics. And the reason they are resistant to antibiotics is because they have evolved strategies for literally deactivating the antibiotics. One way in which conventional antibiotics work is they actually get inside the bacteria and poison the bacterium's machinery such that the bacterium dies. Bacteria, from their perspective, have developed strategies for literally spitting those antibiotics back out. So we have to come up with an entirely outside-the-box approach to killing bacteria that doesn't allow them to develop resistance. And so in collaboration with Hu Dong, an assistant professor in the Department of Chemistry and Biomaterial Sciences at Clarkson, we developed these antimicrobial nanofibers, or antibacterial nanofibers. That's a fancy way of saying these are microscopic needles. So the same way you can take a needle and burst a balloon, these microscopic needles that Hu Dong's group has developed can burst bacteria. and they do so in such a way that they can only burst the bacteria and not the cells that the bacteria are interacting with or infecting. So it's a very effective means of targeting the bacteria exclusively and targeting them in such a way that they cannot develop resistance. The same way a balloon cannot protect itself from being popped if you touch the surface of the balloon with a needle, these microscopic needles puncture the bacteria and they can't protect themselves against it. That's the brilliance of this new approach.